Hello. So as you can tell, nighttime outside. I'm recording this when I most assuredly should not be. I'm very tired, but I wanted to get this done tonight and posted tonight or first thing in the morning so that y'all had all week with it, so that you could study. So our test will be next week, and it will be during your lab time. I know it'll be the first time that we've met in person all semester, but they want these midterm tests taken in person. So it won't be a written test. It will be on the computer, but it will be in the library during your lab time. So my Monday people, is it 4? I can't remember if it's 4 or 4:20. I know that Thursday y'all are 12:20. So we will be meeting in person next week to take this test. Do not forget, I can't let you make it up. No makeups, unless it's Dire Straits. No makeups. See me saying that? No makeups. So they do want us taking them in person. I can't post it. They don't want us posting it onto Canvas and you taking it that way. They want it done in person. Now, if you have any questions about that, please let us know. Please let me know. I will happily answer what I can, or if I can't answer it, I will go find the answer and get back to you. But with that being said, I am posting a blank study guide along in this same module that's going to be titled Midterm Study Guide and Video. Print out the study guide, fill it out as you watch the video. I am going through question by question on the study guide and telling you the answer that goes in the blank. So the test is mostly multiple choice and true false. Mostly 95% multiple choice. A few true false thrown in. Some of the bonuses you'll have will be like essays. You just got to write the answer down. But I found in my education career, as a student, that I did better if I had a fill-in-the-blank study guide. And my high school U.S. history teacher did this for all of her tests. A week before the test, she would give us all a study guide, and it was the questions from the test, and they were all fill-in-the-blank. And so we would go through as a class and fill out the study guide, write down the answer in each of the blanks on the study guide. And then when it got time to the test, if you're studying, if you're studying fill in the blanks and you're given a multiple choice test, you should be good to go. Sorry, y'all. Allergy season is not kind to me and my kids. Usually I'm a coffee girl, but it's 10:40, so it's hot tea. So anyway, with all that being said, print out the study guide and fill it out as we go. So here's the video going over all those questions. So, question 1 on the study guide. In lab 1, the scientific method, we observed the following 3 materials. We observed sodium bicarb, calcium chloride, and phenol red. Okay? That is what we observed. If we had done it in the lab, water would have been in there. So write that down as well. Like with the phenol red, put phenol red / water. But the big ones are sodium bicarb, calcium chloride, water, phenol red. And I know there's only 3 blanks. Just put the water with the phenol red. Since I don't remember there being water in the video that we watched. That's why I didn't put it on there as the blank. But just know that if we had done it in lab, water would have been there. All right. Number 2, the steps of the scientific method in order are observation, So you observe something, you see something happen that you're like, my eyes have seen, my brain has questions. So then you form a hypothesis like, So I'll use my science seminar, for example. When I was a senior, I did my own research. We used to go on a spring break camping trip through the school to Ossabaw Island, which is a barrier island off the coast of Savannah. That is neither here nor there. What I learned while I was there the last time I went was that the natives, the Native Americans who lived on the island, when people started going to the island, they would do these rituals that would last all night, and they didn't have coffee. They didn't have energy drinks. They drank a black tea that was made from the Yopon holly plant, and it was said to give them massive amounts of energy and allowed them to focus on what they were doing overnight. So my science seminar, after I heard that, and I observed that happen, and I was like, my question was, I wonder what's more effective, an energy drink or coffee today, or the tea then? So I looked at what was in like the tea, like what was the energy giving properties, and then I found an energy drink that was somewhat comparable and said, I bet the energy drink doesn't allow you to focus as much as the tea does. I bet they still give you the same amount of energy, but I bet that your brain function is better on the tea than it is on the energy drink. So my hypothesis was, my observation was that, these people use this tea instead of energy drinks. I got questions. So my hypothesis was, I'm betting today's energy drinks will give you the same amount of energy as they did, but it keeps you so like this that you can't focus. So I made predictions, which is the next step. My prediction off of my hypothesis was the tea was going to be better. So then I did an experiment. So we've observed, we've made a hypothesis, we've made predictions, and now we're experimenting. So what I did was I did it in the summer. And so the summer anatomy class all volunteered to be my test subjects. They drank their Red Bull. I took their heart rate and their blood pressure to measure the amount of like energy, like what was their baseline. 15 minutes after they finished their drink, what was their blood pressure and heart rate. And I did that 1 week, or I did that one day, and then the next day they drank it, and then I did an EEG. So I looked at their brain waves while they did math problems, just simple 1 + 1, 2 + 2, 3 + 3, things that you can do in your head. And so then the next week we did the same process, but with the tea. So I did my experiment, collected the data, processed the data, got my results, and I came to a conclusion, which is the last step. After experimentation, you come to your conclusion. So conclusion is the last step. Obviously, it concludes. So if you're wondering, my hypothesis was incorrect, which is fine. That's what science is about. Your hypothesis can be wrong. If you're doing it right, your hypothesis is testable multiple times. You should always be able to repeat the experiment. So my hypothesis was wrong. The EEG showed on the Red Bull versus the tea that it was better with the Red Bull than it was with the tea. Not by much, but there was a significant difference between the two. And when I say significant difference, I'm talking about statistical significant difference. So your p-values and all that fun stuff. I did my seminar in October of 2012. Been a while since I've looked at this, but all that to kind of give you an example of how the steps of the scientific method work. So we observe, we hypothesize, we make predictions, we experiment, and then we come to conclusions, okay? 5 steps, Know them in order. They make logical sense. Just make sure you get them in order. All right. Question number 3. Sodium bicarb and water / phenol red in that experiment create an endothermic reaction. So sodium bicarb was baking soda. So it got cooler. Remember, it absorbs that heat, and it didn't give it off. It kept it. So it was cold. All right. So number 4, an exothermic reaction releases heat / energy. So exothermic is hot because it's releasing heat, or because it's releasing energy. So energy is released and it heats up, so it feels hot. Okay. Number 5, what gas is formed after the combination of the phenol red, calcium chloride, and sodium bicarb? CO2. That carbon dioxide gas. I don't want the acid name, I want the gas name. So it's carbon dioxide, CO2. All right, number 6, what test did we use to determine the presence of proteins? It's the Biuret, B-I-U-R-E-T, the Biuret's test. Carbohydrates, are they organic or inorganic? Think about it this way. Well, just let me tell you. Carbohydrates are organic. Question 8. Starch and glycogen act as reserve storages of food for animals and plants. Okay? Starch is the food reserve for plants. Glycogen is the food reserve for animals. We don't store food reserves as starch in our bodies. Our bodies break down starches and store it as glycogen. So, starch is plants, glycogen, animals. Those are the 2 food reserves. Okay? Glucose is a very important monosaccharide used in cellular respiration. Okay? Important monosaccharide means one sugar molecule. Mono, one, saccharide, sugar. One sugar, glucose. Okay? Cellular respiration, glucose is a very important monosaccharide. Okay? Number 10, Sudan-4 is the test that we use to determine the presence of fats. So, Sudan-4, fats. 4 fats. Okay? Number 11, fatty acids and glycerol. Okay? Fatty acids and glycerol are the 2 main components that make up lipids. So, think about like that phospholipid bilayer. That lipid bilayer in cells. It's fatty. So, fatty acids and glycerol are the key components to lipids. Okay, we didn't really go over the names of these scientists, so I'm going to make sure that you know these because I don't think it's as important, but it's a question that needs to be asked. Robert Hooke was the first scientist to term a cell. What term did he use? Cellulae. Okay, Robert Hooke, cellulae. The next question is another scientist question. Who was the scientist who viewed single-celled organisms found in a pond under a microscope for the first time? So, microscope scientist, Anton von Leeuwenhoek. Again, remember I say this is a multiple choice test. You're not writing this out. So if you see the name compared with the important thing, then circle it and move on. Or click the box and move on. Okay? So the blank is the theory that all living things are composed of cells and that all cells come from other cells. That's the unified cell theory. All living things are made of cells. Yes. All cells come from other cells. You can't have something from nothing. You've got to have something to start with. All cells come from other cells. All living things are composed of cells. They are the building block. All right. The 2 types of cells found on Earth are prokaryotic cells and eukaryotic cells. Okay? Moving on to 16, prokaryotes are single-celled organisms. 17, prokaryotes, they lack organelles, but they have one component that assists in the production of proteins. Protein production. That component is called the ribosomes, okay? Prokaryotes, they don't have organelles, but they have ribosomes to create proteins, okay? Eukaryotic cells range from simple, like unicellular protists, to very complex plant and animal cells. So the majority, I'm not even going to say that because that's not necessarily true. The complex plant and animal cells that we think of and think, oh, with all the rough endoplasmic reticulum and the Golgi apparatus and that kind of stuff, those are the eukaryotic cells. Okay. Oh, number 19. The theory of endosymbiosis explains or attempts to explain how eukaryotic cells evolved. So, symbiosis, you're making it. So, how it evolved, how the eukaryotic cells evolved. All right. The ability to transport. Y'all, this is why you don't do stuff like this at 11. The ability to transport materials across the plasma membrane is important for cellular survival. True or false? If you can't get stuff across that cell membrane, you're toast. So that is true. Okay. Blank is the movement of particles from areas of high concentration to lower concentration. So diffusion. That's diffusion. So you diffuse the situation. You calm it down. You go from high to low, right? High concentrations of anger to low concentrations of anger. Someone has diffused a situation. You diffused a bomb. Diffusion. Particles moving from higher concentration to lower concentration. Okay? The diffusion of water gets its own name. So if water diffuses from high concentration to low concentration, it's called osmosis. So you can't sleep on a book and learn. That's not how that works. Osmosis is the diffusion of water. Okay? In a blank solution, the cell shrinks because the water flows out of the cell. So if the cell shrinks, the solution is hypertonic, right? See last video on tonicity if you have questions on that. All right. Number 24, plasmolysis. Plasmolysis is the process of which when cells shrink at a certain rate, it causes a space between the cytoplasm and the cell wall. So plasmolysis, like the cells are shrinking consistently at a certain rate, and there's a space between that cytoplasm and the cell wall. Okay? Number 25, what stain was used in lab 3 to observe cheek cells under the microscope? Methylene blue. That's why they're blue. So methylene blue was the stain used in lab 3 to observe the cheek cells under the microscope. Okay? These two organelles are the organelles that function in the support of the cell. So the cell wall and the cytoskeleton. Makes sense. Think about it this way. A wall holds up the house, right? Your skeleton in your body holds you up. So the cell wall and the cytoskeleton function to support the cell. All right. Protein synthesis is the function of what organelle? So that's the rough endoplasmic reticulum. All right, 28. The mitochondria is essential for supplying energy to the cell through the synthesis or the making of ATP. You can't function without it. That's our energy. That's how we live, ATP. All right, 29. The two main processes that drive life in plants and animals are cellular respiration and photosynthesis, okay? Photosynthesis is the plant version of what cellular respiration is for us, okay? Photosynthesis takes an inorganic carbon and water to create energy in the form of glucose, okay? So photosynthesis takes the carbon and the water, so the inorganic carbon, that's important, inorganic carbon and water, and it creates energy that's usable for us in the form of glucose. So, 31, what is the chemical equation for photosynthesis? So, time.
No.
Please tell me I have a marker over here. I am so thankful that my daughter has an affinity for drawing on whiteboards. Because I have one. She's 4 and left-handed. So when she draws on a whiteboard, it erases immediately because, you know, lefties. If you're a lefty, you know how that goes. All right. So the chemical equation for photosynthesis is you have 6 CO2 plus 6 H2O. Okay? So 6 carbon dioxides plus 6 waters. And that's going to yield you, let me write it and then I'll flip it around. C6H12O6, which is glucose, plus 6O2, which is oxygen. So, this is what's so cool about photosynthesis, okay? So, it takes our byproduct. So, when we breathe out carbon dioxide. So, it takes 6 carbon dioxide plus 6 water, and it creates things that we can use, so glucose and oxygen, okay? So, I hope that y'all can, hope this is not backwards. So, it's 6 CO2 plus 6 H2O yields C6H12O6 plus 6 O2. That's photosynthesis, okay? Now, the next one we didn't go over at all, but it should be in something that y'all read in the process of studying. So, blank is a key carbon-fixing enzyme in the process of photosynthesis, and it functions optimally when the pH is 8. So, 8's a little higher than baseline, which is like 7-ish. So, we know higher pHs are bases and lower pHs are acids. But this thing is finicky. So, it's called Rubisco. R-U-R. Punctuation or capitalization matters. So, it's ribulose 1,5-bisphosphate carboxylase oxygenase. So, Rubisco. Ribulose 1,5-bisphosphate carboxylase oxygenase. Rubisco. And it is the key carbon-fixing enzyme. It ends in ACE. It's usually an enzyme. In the process of photosynthesis. So it fixes that carbon where it needs to be. And it functions optimally at a pH of 8. All right. So, question 33. In cellular respiration, glucose is used as the sugar molecule for the energy source. So, remember a while ago when I talked about that byproduct of photosynthesis where we had 6 CO2 plus 6 H2O and it yielded C6H12O6? That's glucose. And animals take that and undergo cellular respiration so that we can have energy to live. Okay? So, glucose, sugar molecule, energy source for cellular respiration. That's 33. All right, 34. Phenol red was the solution used in lab 4 to indicate an optimal pH level that showed photosynthesis was occurring. All right, 35. This is the question about Rubisco and how it's finicky. The Rubisco enzyme will stop working altogether if the pH reaches 6 on the low side and 10 on the high side. So it really is optimal at 8. It lacks that more neutral area of the pH scale. Okay, next is true, false. Photosynthesis can only occur in plant cells. Yes. We don't have the proper stuff to do photosynthesis. You got to have chlorophyll, and we don't have that. That's a plant thing. Okay? All right, let's see. This next question we've already gone over, but I'm going to do it again. Glucose is a monosaccharide. That means it only has one sugar molecule. Monosaccharide, one glucose. Very important, one. Okay? All right. So we talked about photosynthesis in the equation, this chemical equation. All right. So now we're going to look at the chemical equation for cellular respiration. So it's kind of flipped. So take my husband's biochem class. You're really, really going to break this down in minute detail, which is important stuff to life. Just throwing that out there. So, all right. Remember, we had on the cast off of photosynthesis, we had C6. Y'all can't read that. Let's go back to black. My other markers are better on this board, but where are they? That's the downfall of having a daughter who has an affinity to drawing on marker boards. I can't keep up with a marker. All right, going back to black. All right, so remember we had C6H12. That's better. O6. All right, so there's your glucose. Plus, we had 6 oxygen gas. So, glucose plus oxygen. Remember, this is cellular respiration. Question number 38. So then we're going to yield 6 waters, 6 H2O, plus 6 CO2, plus ATP energy. So you've got 6H. So, 6. These are not my favorite markers in the world. Sorry, guys. Trying to make this a little darker. They're fine point and Alright. So you've got glucose plus oxygen yielding water, carbon dioxide, I'm not writing that, and ATP energy. So see how it's just kind of the flip it and reverse it here, pulling a Missy Elliott, flipping and reversing it. We're here for that. Y'all are probably too young for that reference. It's totally fine. All right, so glucose plus oxygen yields water and carbon dioxide and energy, ATP. Energy that we use, because remember, this is cellular respiration. Oh, you can't read that. Cellular respiration. C6H12O6 plus 6O2 yields 6H2O plus 6CO2 plus ATP. Good? Good. Okay. Put this down. I think that covers the whiteboard. I hope it's not backwards. It probably is, though. I'm doing this on the front camera. All right. Okay. Make sure that didn't fall off the table. All right. Number 39. Key components for photosynthesis. Light and chlorophyll. Remember I said that. You got to have that chlorophyll or it ain't going to work. And light. Photosynthesis is not going to happen in the dark. Got to have that sunlight. Got to have that light. Those are the components of photosynthesis that are needed / yielded along with carbon dioxide and water in order to reach your final product in photosynthesis. All right. In lab number 4, we observed yeast cells undergoing cellular respiration. Eukaryotic cells are multi-celled organisms. Multi-celled. Number 42, methylene blue is poking its head back out to say, hey, that's what you use to observe the cheek stains, the prokaryotic cells, and the eukaryotic cells. So methylene blue was used to observe all 3. Okay? Sorry, guys. My contacts are killing me. Number 40, that was 42. Okay, 43. Photosynthesis occurs in a certain organelle, and that's called the chloroplast. Okay. The site of protein synthesis is the ribosome. Cellular respiration converts glucose to ATP, which is what we use for energy. All right, 46. Photosynthesis is a multi-step process that begins with water and an inorganic carbon. Inorganic. I-N. Not organic. Inorganic carbon and water. All right, 47. Lysosomes. So if something gets lysed, it starts getting broken down. So it breaks down old organelles and plays a role in cell death. All right. 48. Carbon dioxide is a main requirement for photosynthesis. In the lab, we observed sodium bicarbonate that was used for a source of carbon of photosynthesis. So in that photosynthesis lab, if we had done it in lab, we would have used sodium bicarb or baking soda for that carbon to do photosynthesis, okay? Cellular respiration uses glucose as an energy source. And know how to calculate total magnification. If I were to say the total magnification of a scanning power lens, what would it be? 40. 40 times 48. Okay? That's all 50 of the questions on the study guide. I will give bonus questions. Some of them may be related to what we've done. Some of them may be related to other stuff that are really easy to answer. I don't know yet. I haven't put them on there. All right. Probably 10 points worth, like I said. So roughly 5 questions. This test is 50 questions, and it's worth 100 points. So it's 2 points a pop. 2 points a piece, 100 questions, or 100 questions, 100 points, 50 questions, okay? Let's see. Like I said, we will be meeting in person to take this test in the library, okay? 100 points. Watch this video. Kind of fruitless for me to say that at the end of the video. I'm just reading off my notes that I had. If you have any questions, please reach out. If you have any questions about when, where, how we're taking the test, please reach out. I'm happy to answer any that I have. Tell me what lab section you're in. Those of you on Monday, again, I would just, if one of you wants to double check me on the time, I'm 95% sure your lab is scheduled at 4 o'clock. I know that sounds terrible, but I can't. I've got 42 different times in my brain, and right now I'm not able to recall the time that y'all's lab starts to the top of my brain. I just can't remember if it's 4 or 4:20. I know 12:20 is Thursday. And my schedule for some reason it's not letting me into it on my portion of Scholar. I'm trying. I really am. Every time I look, it's like, page broken. I'm like, no bueno, because I need this like yesterday. We'll try one more time while I got y'all here. Okay. Let's see here. Okay, so it's 4:30. Yay. 4:30 to 6:20 is y'all's scheduled time on Mondays. So we will have the lab at 4:30. So, the computer lab. So, we will be taking the test in the library computer lab at 4:30 on Monday, October the... I can't look at the calendar on my phone because I video these on my phone. For some reason, my computer doesn't want to work with me. Monday, October the 9th. Will be when we take the test at 4:20 in the computer lab for my Monday labs. And then Thursday, October the 12th at 12:20 in the library computer lab, we will be taking the test. If that changes, I will announce it on Canvas. If that changes, I will announce on Canvas that that is changing. I doubt it changes, but I just need you to know, okay? All right. Happy studying. Again, questions, comments, concerns, doubts, fears, anxieties, you reach out to me. We'll get it taken care of, okay? Have a great day, week, month, year, and I'll see y'all next week. Bye, guys.